My name is Dee Taylor. I'm Corporate Director of Research for Interior Health, as well as Co-Scientific Director for the Rural Coordination Centre of BC. I am privileged to be here today to be moderating this session, and really excited to hear from three amazing speakers. Dr. Alison Kelvin will be giving a presentation titled Introduction to Immunity. Dr. Darby Thompson will be speaking to us about the strength of data through the phases of clinical trials. And Dr. Manish Satarangi will be speaking to us about COVID-19 immunity. All three of these speakers are amazing, and I encourage you to check out their bios in the program. I look forward to hearing from all three, and then after the presenters are completed, we'll have an opportunity to ask questions. So welcome to this session, and take it away, Dr. Kelvin.
Hi, thanks so much for having me. My name is Alison Kelvin, and I'm an assistant professor at Dalhousie University and a scientist at the Canadian Centre for Vaccinology and at Vito Intervac in Saskatoon, Saskatchewan. I'm going to tell you a bit about my work on vaccines for SARS-CoV-2 and COVID-19 and where we are now in the development of vaccines. So the title of my talk is Emerging Viruses and the Need for Flexible Vaccine Platforms, the COVID-19 vaccine story. So just to give you a bit about myself, I am an emerging virus researcher, and the goal of my research program is to understand the pathogenesis of emerging viruses to develop vaccines for these viruses as they emerge. I opened up my laboratory in Halifax, Nova Scotia at Dalhousie University in the IWK Health Centre in partnership with the Canadian Centre for Vaccinology. But after SARS-CoV-2 emerged last January, I then relocated to Saskatoon, Saskatchewan to use their CO3 facility, which is a high containment facility able to provide the proper infrastructure for working with SARS-CoV-2 and high containment viruses. So the goal of my research lab, the ultimate goal, is to develop vaccines to protect people against emerging viruses that cause severe illness or death. And to go into a bit of background on emerging viruses are typically viruses that spill over from animal reservoirs. And with these viruses have the potential to cause infection in people, and then even more potential to spread throughout human populations. This is the definition of emerging viruses. And although we can't predict exactly which virus will spill over and emerge, we do know some of the viruses that are more threats for causing spillover events and eventually possibly leading to pandemics. And these are the focus of my research program and should be the focus of certain vaccine development. So some of these important viruses that we have been watching out for include filoviruses such as Ebola, influenza viruses that may emerge from avian species or porcine species, as well as coronaviruses that have an animal reservoir which we believe to be bats. So if you look in the media often has these dramatic headlines of emerging viruses that all have this common thread of what is the truth about SARS, the truth about Zika virus. And when we really get down to it, the truth about emerging viruses is that there are many unknowns. And these unknowns become our research priorities. So going through these unknowns and starting with the clinical picture, we all often don't know what the clinical picture looks like as people start to be infected with these emerging viruses. We don't know who the susceptible populations are. We don't know the animal origin of the spillover event to stop future spillovers. And we don't have a preclinical model. We don't understand transmission after these viruses first emerge. And importantly, we often don't have therapeutics or vaccines to prevent disease. And this is really the focus of my talk today, is how do we develop vaccines that can be flexible and quickly established in the event of a virus spillover. So personally, I began working on emerging viruses in 2003. This was shortly after I finished my undergraduate degree. And I was working as a summer student in Toronto. And as cases were coming into the hospital networks, I was involved with the SARS relief efforts in two ways. One, I had a research project that was dedicated to understanding why certain people who were infected with SARS became severely ill and died. And the second part, the hospital networks got all the summer students to work as screeners as patients and hospital staff came into work. And it was really interesting to be involved in both the social aspects of disease prevention and also the research aspects of disease prevention for SARS-CoV-2. Since then, I have worked on many of the major emerging viruses. Specifically, I've worked on chikungunya virus that caused a major outbreak in Italy in 2007. I worked on understanding avian influenza viruses in 2008, which really became a platform that was easily pivoted when the swine flu H1N1 pandemic came out in 2009. I was one of the first groups to identify the pathogenic mechanisms of H7N9 avian influenza when it emerged in 2013. And finally, I worked on Zika virus in 2016. And through all of these, what the common thread here is that we need quick development of vaccines to stop a pandemic. It's been estimated that we'll need to have scaled up human doses within 4 months of an emerging virus spilling over to stop a pandemic. So in my lab, I have 3 major research projects. One with influenza being the core of my research because it's the constant emerging and re-emerging virus. My project 1 focuses on the immune responses to influenza viruses. Second, my second project focuses on the mechanisms of vaccination so that we can search for a universal vaccination or a vaccine that's easily flexible during a spillover event. And third, my third project focuses on emerging viruses and pandemic preparedness. So really what this means is my first project is why do we get sick? The second, can we make a vaccine for that? And both projects really feed into having the tools and the knowledge ready for the next pandemic threat. So on the evening of December 30th in 2019, I was going to bed and I checked my email one last time where I had an email from ProMed Mail, which I'm sure you all know to be kind of like a social network for infectious disease researchers. And this email described an outbreak of atypical pneumonia in Wuhan, China, that led to the hospitalization of almost 30 people. As you read through the email also suggests that hospital networks become prepared to identify patients as they present themselves in certain healthcare settings. So I had some immediate thoughts and responses. The first one was that either everyone who was infected with this possible new virus had severe disease, which led to hospitalization and possibly death, which was a major concern because then we were dealing with a high pathogenic virus. The second scenario was possibly that this virus was widespread in the community. And like with influenza, the number of hospitalizations that we see are only indicative of a small proportion of who's infected, the most severe cases, which meant that the virus might be more widespread in the community. So I use this iceberg as a representation, with what you see on the surface of the water being the hospitalizations, but underneath the surface being a large number of mild or asymptomatic cases that aren't being accounted for through the number of hospitalizations. So this was quite alarming to me when I considered these two scenarios. One meaning that we had a high number of people actually infected and possibly person-to-person transmission. So I put together a strategy to start working on this virus and developing a vaccine. First, I knew we needed to acquire the virus. Second, I knew I didn't have a CL3 to actually work on the virus. And this is when I reached out to Vito Intervac and spoke with Daryl Falzerano to acquire the virus and develop a preclinical animal model, which was the third aspect I needed for vaccine evaluation or vaccine candidate evaluation. And four, we needed to start designing vaccines based on platforms that we had already developed. So when we develop a vaccine, there's a number of steps that often takes several months to years, as I'm sure everyone in the audience knows. But the first, when we have an emerging virus, our first step is identifying the target. We had to figure out what the virus was, and this came after sequencing of the SARS-CoV-2 virus in early January. And on that virus, we needed to identify the target, where the vaccines would be developed from. And most of these targets for the SARS-CoV-2 vaccines in development are based around the spike protein that's located on the virus envelope. Once we have a target identified, we can then start develop the vaccine platforms, which there are 4, and I will go into in the next slides. And then this can lead to preclinical evaluation of these vaccines. So we needed to identify an animal that was susceptible to the virus and could produce a productive immune response after vaccination, so that we could evaluate, was the vaccine protective against infection and possibly the development of COVID-19, the disease. Once we've identified a promising candidate, the vaccine must be scaled up for phase 1 and further human clinical trials. If we look at our timeline, we identified the virus in early January in 2020. We started developing vaccine platforms January to February after the sequence was released, which led us directly to start putting together preclinical evaluations and developing a preclinical model and scaling up for phase 1 clinical trials. And then of course, phase 3 clinical trials, we started getting some data for those in December. So there are 4 different types of vaccines. The first one being nucleic acid vaccines, and these are essentially either DNA or RNA encoding the target, the viral target. Protein-based vaccines, which might be a protein or protein subunit of the virus, or a virus-like particle. The third being the actual virus, and of course these were the first vaccines where we took viruses that were infecting people. And we either downgraded them or attenuated them, or we inactivated them with chemicals or other methods. And then the fourth method being a viral vector. And this is kind of using a different virus that has been also attenuated as almost a Trojan horse for bringing in our viral target for our specific virus. So now I'll just briefly explain the differences between these vaccine platforms. So nucleic acid-based vaccines are pieces of DNA and RNA and are currently in development or now being used for SARS-CoV-2. The benefit of the nucleic acid-based vaccines is that they're easy to produce and fast, which is great for a pandemic threat. Preclinical data has shown that that RNA-based vaccines can produce both a type 1 and type 2 T-cell response. The drawback of these vaccines is that RNA degrades quite easily, but the new technology has been able to from Moderna and Pfizer, has been able to keep these RNAs relatively stable. And the problem with DNA-based vaccines is that the DNA doesn't always get into cells. And here you can see in this diagram where lipid nanoparticles are delivering a piece of RNA into cells, where the cells can then use the instruction from the RNA to produce the protein, such as the spike protein, which then on the other side can elicit an immune response against that specific protein. So the major nucleic acid-based vaccines, and 2 of them we're lucky to have approved here in Canada, include the Moderna for RNA, it's the Moderna and Pfizer vaccines. These are both based on a pre-fusion stabilized spike protein. Vaccination in volunteers has shown the production of neutralizing antibodies, and again, both of these are approved for interim use in Canada. DNA-based vaccines, the main one that has been in development for SARS-CoV-2 has been the Novio vaccine. And to get around the problem of getting DNA into cells, Novio uses this electroporation gun, which possibly might not be the easiest way to vaccinate people. So the second type of vaccines is the protein-based vaccine, and here we can see pieces of the virus being broken down into spike protein, and this is either made by breaking down the virus or by producing these proteins in vitro. And they're a common vaccine platform. The benefits is that they focus the immune response on possibly a single protein or a number of proteins. And the drawbacks is that because we're just vaccinating with a protein, we might need an adjuvant or a prime boost schedule. And developing these pieces of protein might take a long time, which is another one of the major drawbacks. So the main contender for SARS-CoV-2 vaccines right now in the protein vaccine type is a Maryland-based company named Novavax, and they've developed a stabilized pre-fusion S protein that's kind of creates these microparticles, and it's adjuvanted with matrix M, and currently their COVID-19 vaccine is in phase 3 clinical trials. Importantly, the Novavax vaccine has shown high levels of neutralizing antibodies in participants. Our third type is the viral vectored vaccines. This is another really important vaccine platform. And again, we're using a different virus that has been attenuated or somehow genetically modified to not induce disease, to carry either the full protein or the instruction for a protein on our target virus. So there's a couple of adenovirus viral vectored vaccines in development right now that carry instruction for the corona spike gene, which after the virus, the vectored virus infects our cells after vaccination, our cells can then start producing spike protein similarly as is done for our nucleic acid-based vaccines. The benefits of this is that viral vectored vaccines will elicit a robust immune response because we're using a virus to actually deliver our target. But the drawbacks is that these types of vaccines are difficult to develop. They're also laborious to grow and purify, and our immune responses might be counteracted because of pre-existing immunity, or we may develop immune responses directly to the virus being used as the vector instead of the target as is intended. So our major contender for virus vectored vaccines is the AstraZeneca vaccine, which was developed in part with the University of Oxford. This vaccine has been approved in the UK, and it's based on a chimpanzee adenovirus that carries the coronavirus spike protein, or instruction for the coronavirus spike protein. This platform was previously developed for MERS, influenza, TB, chikungunya, Zika virus, meningitis, and the bacteria that causes plague. Preclinical studies has showed reduction in respiratory inflammation in a preclinical model, and we know that this vaccine can elicit antibodies in preclinical models as well as humans with a balanced Th1 and Th2 response. So our last type of vaccine that I will talk about is protein vaccines, or the VLP virus-like particle vaccine. And basically a VLP virus-like particle looks like a virus, but it has no nucleic acid in the inside. So it has no genetic instruction. It's kind of like an empty virus that's used to elicit an immune response in our bodies after vaccination. So the benefits of this is that it has the necessary virus structure to elicit a strong immune response, but the drawback is that it might be lengthy to produce and purify. The major VLP vaccine that's being produced is from Medicago right now, which is our Canadian-based vaccine. So what does our Canadian vaccine landscape look like? I've spoke about many of the contenders, which are more international contenders. I also wanted to mention what's going on here in Canada. We have a DNA-based vaccine from Entos and Epivax, a peptide-based vaccine, which is being developed by IMV, or Immunovaccine out of Halifax, Nova Scotia. Dr. Chris Richardson at the Canadian Centre for Vaccinology has developed a viral vector-based vaccine based on the VSV virus. And this is the same platform that was used for the Ebola vaccine. Dr. Daryl Falzerano at Vito Intervac has a peptide-based vaccine that's based on spike. And then finally, Medicago, as I mentioned earlier, has developed a VLP-based vaccine. So this empty virus that has no genetic instruction, but can elicit a strong immune response because it has the structure of the virus on the outside. So why do we need multiple platforms? Well, to vaccinate the entire world as quickly as possible, we'll need all these platforms working together to get vaccine doses to all the people who are in need. And with that, I'd like to thank you. I'd like to thank everybody in my lab for the work that they've been doing on SARS-CoV-2 model development and vaccine evaluation. And I'd like to thank Vito Intervac for hosting me and giving me a space to work on this high containment virus. Thank you very much.
Hi everybody, my name is Darby Thompson. This is my dog Chip. She's a Yorkshire Terrier. She's 4.5 pounds, and I wanted to show her to all of you because I wanted to explain the barking sound you will almost certainly hear during the course of this video. And you may see her come up again during the presentation. So during this middle talk of the middle day of a virtual conference, you might have reason to watch further. So we'll send Chip on her way and get going. So today I'm going to talk to you about the strength of evidence in vaccine clinical trials. Before I get going, I want to make sure that I thank all of the first responders, all of the frontline healthcare workers who are on the front lines dealing with this pandemic. Their sacrifices are appreciated beyond words. But I also wanted to acknowledge the many people who are putting themselves at risk, working in grocery stores and home delivery and other areas of the service industry that are putting themselves at risk to allow us, to those who are privileged to be able to work at home and stay at home, to do so. And I hope that those of us who are able to continue to avoid putting ourselves and others at risk out of respect for those who have no choice. There are three core takeaways from today's talk. The first is that the strength of evidence in vaccine trials is extremely content specific. There are not fixed thresholds for every disease being studied, or for every vaccine, or for every population, or for every endpoint. The second is that the transparency of the vaccine development process, the clarity with which the way the evidence was gathered and compiled can be presented, and the inclusivity of all the populations at risk make the evidence that we collect more accessible. And the third is that partnerships, platforms, and infrastructure have enabled expediency. There is no way that the vaccines for COVID in 2020 could have been developed without public-private and private-private partnerships, with the open sharing of data and technology and a shared and common interest. There's no way we could have developed vaccines this quickly. And I think it lays the groundwork for a future of better collaboration. Vaccine clinical trials are developed very much in a similar classical framework as most drug and therapeutic products. Phase 1, 2, 3. In phase 1 always begins with healthy volunteers, healthy adult volunteers. That's not necessarily the target population, nor where the product will end up being deployed, but it is where all vaccine studies start. Phase 1 is usually conducted, as with therapeutics, in a very controlled setting, and it's really to understand safety and tolerability in tens of subjects. There's not usually a statistical element to design of these. Phase 2 is where we get into randomized controlled studies in order to identify the exact dose frequency regimen that you're going to carry on to phase 3. There is some statistical component to phase 2 in determining how many subjects you need to confirm immunogenicity, depending on how many arms you need to evaluate, and in order to rule out high frequency safety events. The real element of phase 2, and often the crux, is that you're characterizing the immune response. It should be as expected, as designed. If you see some sort of immune response, but it wasn't the one you're predicting, you're often going to need to go back and re-engineer, re-understand, potentially restart, dog number 1, restart the phase 2 trial. Phase 3 is what we're most familiar with in terms of large-scale randomized protective efficacy trials. That these are often done in the exact target population or subpopulations that are of interest, and they're sized to adequately estimate efficacy and safety. Now, there are many variations of the sorts of designs that I just talked about, a number of alternatives. That was sort of a Goldilocks pattern, going from 1, 2, 3 in order. And as it happens, most of the COVID vaccines have been developed in essentially a Goldilocks pattern when we speak of like the Moderna or the Pfizer BioNTech vaccines. In the design of the trials, there's lots of adaptation is common, sample size re-estimation, enrichment designs, interim analyses. The Pfizer vaccine was a seamless phase 1, 2, 3 trial. So there's variations of these. You might be doing a stepped wedge design or a cluster randomized design if you don't have the same sort of broad-based public health requirement that you do in like the COVID pandemic, or in other peripheral indications, subgroups, alternative endpoints. You may be supplementing components of trial data, potentially parts of failed phase 3 trials, with observational data that you've gathered from public health databases. So there are many variations of these stages, but that's the phases. The 3 phases are the core. You'll notice I've left off challenge study entirely. I tried to find a way to talk cogently about challenge studies in this presentation. It's just not enough time. If you are interested, please ask me in the discussion. We could talk all day about challenge studies, and I'd be happy to do so, but it doesn't fit into this topic today. For those of you who are interested in learning more about vaccine development, this is a very nice review paper by Singh and Mehta. It's not in a super high impact journal. It's the Journal of Postgrad Medicine, but it's an outstanding paper. It's referenced in a lot of other literature, and it gives you a really nice overview of the phases of development, along with some very specific examples of product development, some of which followed the Goldilocks pattern, the phase 1, 2, 3, others that had a more circuitous route to getting approval in specific subgroups. So to reiterate, you're going to always size, primarily phase 3, but throughout, you're going to size a study to confirm safety and estimate vaccine efficacy, but you need to do this in the target population and important subgroups. You might think of that in the COVID setting as we need the broad-based vaccine protective efficacy of the vaccine, but we also want to know specifically, does it work well in individuals in the 20 to 30 age group who are carrying most of the infection and are most likely the primary vector for its spread, and are we interested in the protective efficacy in those who are 65, 70 years or older, who are most likely to progress to severe disease or die? To speak specifically of the level of evidence and safety, and to contrast vaccine development with therapeutic development, vaccines generally are going to be developed to be deployed on a population level. So when we're accumulating data throughout the trial process, we are seeing events and safety issues as they occur. We're able to quantify those and compare them to those events that are occurring in the placebo group, and experts can be making cogent assessments of the risk of a product. But on a population level, we're going to be exposing many, many more people to the product than we'll ever evaluate in a clinical trial. So key is to estimate the maximum likely incidence of events that we haven't seen. And as you go through the development process, you are accumulating subjects and getting more and more confident that events you haven't seen truly do have a low incidence rate. So in phase 1, you're effectively ruling out events that are going to occur with an incidence of 15, 20% down to maybe 3%. Events we don't observe, we're pretty confident that they don't have an incidence higher than 0.1 or 10%. In phase 2, you're going from dozens to hundreds, maybe even thousands in a very large phase 2 trial. And you're then effectively ruling out events with an incidence of between 2.5 and around about 1 or maybe 0.5%. Phase 3, we get into the much larger trials where we're getting up into the thousands, and effectively you're ruling out events that are fractions of a percent. Specifically, I'll pull out one point. 3,000 subjects would give us high confidence that we haven't missed an event with a prevalence of more than 0.1%, or that events we haven't observed, I should say, won't have a prevalence of more than 0.1%. I didn't just pull that number out of thin air. Here is the guidance for industry from the FDA on COVID vaccine development. This came out in June 2020 before the phase 3 trial started, and they've explicitly said that the pre-licensure safety database for preventative vaccines for infectious disease typically consists of at least 3,000 study participants vaccinated with the dosing regimen. Most vaccines are going to rule out prevalence of events of more than 0.1%. They've explicitly said that they expect that COVID trials will have far more subjects than that. And that's what we get out of the large sample size for safety. We are better and better characterizing the rate of events we do see and continuing to rule out the likelihood that there will be other things that we have not yet seen. Now, so that covers sort of how you would size and the level of evidence that you're accumulating over the trial process with respect to safety. There's also efficacy to consider. And in this case, for COVID, the FDA has given a very clear threshold on what efficacy has to be. To ensure that a widely deployed COVID-19 vaccine is effective, the primary efficacy endpoint for a placebo-controlled efficacy trial should be at least 50%. They also qualify that by saying the statistical precision of the vaccine efficacy must exclude 30% below. So not only do you need the vaccine efficacy point estimate to be high enough, you need it to also exclude 30% below. You need a large enough sample size to exclude 30% below. Let me pause there briefly and just touch on vaccine efficacy. It's thrown about, you've heard the word, I guarantee you, many, many times, and not everybody gives a very good definition. You sometimes hear that 95% of people who get the vaccine will be protected. That's not what it means. Vaccine efficacy is a relative term. It means the reduction in risk of infection if you're vaccinated relative to not being vaccinated. I'll give you a specific example from the Pfizer-BioNTech vaccine publication, the primary results publication that came out late December 2020. Here you can see on the y-axis is the number of lab-positive COVID-19 cases. They had 162 COVID cases following the second dose and in the control group, and 8 in the vaccine group. So the excess cases in the control group is 154 more cases in the control group compared to the vaccine group. The vaccine group has reduced the risk by 95%. To contextualize that 95% vaccine efficacy, it's comparable between the Pfizer-BioNTech study and the Moderna study. But in context, the seasonal flu vaccine from 2018, which is actually a trivalent vaccine, was only 25% vaccine efficacy against the seasonal flu. Had 67% against H1N1 and 42% against all other B type as trivalent. It's an approved product that doesn't even meet necessarily the thresholds that the FDA defined for COVID. That's again partly related to the number of people who become infected. It's partly related to the infectivity of influenza, and it's partly related to the fact that they kind of get one swing at the seasonal flu, because by the time they've built the vaccine, the season's almost over. You don't get a chance to get a second swing at it. HPV, 95% effective. The classic MMR vaccine that's now 50 years old, 60 years old, coming up at its 60th anniversary, after a single dose, 93%, 78%, and 97% effective against measles, mumps, and rubella. Now, the latter two rows here represent two of the major pandemics that we've been struggling with either for decades or millennia. Malaria still kills hundreds of thousands of people every year. Just 2 years ago, it killed almost 1 million people, almost 900,000 people. The best malaria product that has completed late-stage efficacy testing was between 20 and 40%, had between 20 and 40% vaccine efficacy. Nowhere near what we can see in a coronavirus vaccine. HIV, again, the latest, the best late-stage product that's completed testing was 30% effective. That was the TIE study using AIDS-Vax, Alvac. I worked on that trial, and the overall study was about 30%. Various sub-analysis gave very differing results. It was inconsistent, and later sub-studies could not even replicate the 30%. But in both of these, one might say, I understand how many people are dying of malaria, getting infected, and dying of HIV/AIDS. These convey some protection. Why are they not approved? Well, it's because there's more than just the vaccine efficacy and the safety to play into this. There are major public health concerns. There are health economic concerns. If you think of a 30% effective HIV vaccine, if you consider the cost of rolling out, producing and rolling out and administering a vaccine to a large population, it is substantial. Whereas could that money be better spent in directed public health campaigns, both at the population level and in the subpopulation of those who are at higher risk? Protection from malaria at 20 to 40% is better than nothing. Why aren't we rolling this out? Well, there's other ways to approach malaria. A, it can be treated. B, there's bed nets. C, there's topical treatments and other mechanisms you can undertake to prevent the bite in the first place. And the cost of those is orders of magnitude cheaper than the cost of development, manufacture, deployment, administration, and monitoring of a vaccine, which could have adverse events. There are very few adverse events from a bed net. So all of these efficacy results have to be taken in context with the extent of the epidemic, the infectivity, and other considerations, including health economics. There are a couple of groups in both the United States and Canada that are tasked with determining and making recommendations about use based on the level of evidence available from clinical trials and other sources. In Canada, that is the National Advisory Committee on Immunization, the NACI. All their recommendations are publicly available on their website, and as is their process for making the recommendations and the evidence they choose to consider. The ACIP, the Advisory Committee on Immunization Practices at the CDC, has a similar published handbook about how they utilize evidence, what they can use for evidence, how they score the evidence, and how they weight the evidence and inform the decisions about broad-based population immunization programs. And again, All of the evidence for every vaccine they reviewed is available publicly on their website, where they summarize all the information that they used in order to make their justification. They use something called a grade process that scores every endpoint for how important it is and how certain they are in the outcome. Here is the publicly available grade summary for the Moderna vaccine. You can see the first row here is the symptomatic laboratory confirmed COVID-19. They have one clinical trial to support that endpoint. Now, we all know they don't have one, they have several clinical trials for that, but only one was a randomized prospective phase 3 trial that they really felt provided sufficient evidence for this. The certainty column, they have type 1 high certainty that this randomized controlled trial was able to reduce the risk of symptomatic laboratory confirmed COVID. If you go down a couple of rows down here to all-cause death for exactly the same clinical trials, they have very low certainty. And you can see here that is because of imprecision and indirectness. Those two things combined are essentially to say that they've seen very few events. You actually can see in the same table, out of the 30,000 people, more than 30,000 people that have been enrolled in this trial, only 13 have died, which is good, but there's not enough information there to say that even though we're stopping the spread of the infection, that we're actually reducing the death rate. And here, in this context, at this stage of the pandemic, the level of evidence that was required to recommend usage is really only the stop of the spread. They have additional safety data that is summarized in other places that shows that there's very little difference between placebo and vaccine, but with respect to specific endpoints, only the stopping the spread is important. Whether we can actually show that they've reduced the death rate will be really challenging because it's so low. The grade evidence and categorization, as you can see, that from the Moderna and the Pfizer vaccine looks very similar. There's one big phase 3 trial that supports the infectivity endpoint and a number of other endpoints. The AstraZeneca work was their primary defined analysis was doing multiple phase 2 trials throughout the world in different populations and pulling subpopulations together into a meta-analysis. Not only did that not show great efficacy, it was really difficult to see and understand what the true efficacy in a population was with the subgroups and the way things are combined. So there we get to the transparency and the clarity of how something was conducted. A large-scale parallel group trial is easiest to understand and easiest to explain. We actually see the very similar thing in the malaria trial I gave the example of, and in the HIV trial. There are many subgroups, there are many ways to analyze the data. The clearest you can be, the clearest you can define your population, the more accessible that data is, and the higher the likelihood not only of approval, but of large-scale implementation of an effective product. So the future of vaccine trials in general, I think, as I said, transparency and inclusivity is reiterated as the key for success. We'll probably move away from this in the decades to come, but we can see explicitly in COVID the trials that were clearest, the trials that included the most people, that included the most stakeholders in their development, that included the most partners, were the most likely to succeed. These private and private-public partnerships laid the foundation both for infrastructure and for execution of these trials. Large-scale clinical trial networks were able to be engaged to execute these trials expertly and quickly. You can see that sort of breakdown occurring again in the AstraZeneca trial, where they had dosing problems, language barriers. They had a lot of challenges because they didn't have an existing strong infrastructure to deploy the trial in a certain way. Platform trials and/or trial infrastructure enabled acceleration of treatments. And again, we're going to see that this investment in these networks, these arrangements of people who can execute trials together, really lay the groundwork for some of the things you're hearing in this symposium about real-world clinical trials. Having a population of people willing to undertake research, to volunteer for research for a common good because they understand it, is really critical to effectively developing, deploying, and relaying evidence about not only vaccines, but a lot of clinical research. If you have any questions at all on this or any other element, I'm happy to provide all my references. You can email me at dthompson@ms.com. And thank you very much.
Good afternoon. My name is Dr. Manish Sadarangani. I'm a pediatric infectious disease physician scientist at BC Children's Hospital in Vancouver. I'm director of the Vaccine Evaluation Center, where I conduct translational vaccinology research. I'd like to thank the organizers for asking me to come and talk to you this afternoon about COVID-19 immunity and how that relates to vaccines. These are my disclosures. And so the outline of my talk, I'm going to briefly touch on the original rationale for the basis of COVID-19 vaccines using the spike protein. I'm going to look at what we know about the immune responses to COVID-19 infection and look at the current vaccines and try and correspond what we have with efficacy data and immunologic data. So the basis for COVID-19 vaccine development has really been this spike protein, which is shown on the right-hand side here as the red protein which decorates the surface of the virus, and as most of you will know, is the way the virus interacts with the host cells. So here, shown below, is in red the spike protein on the viral surface interacting with the ACE2 receptor on the host cell. And most of the vaccines that have been developed, particularly those that have used the genetic engineering methods, whether that's the DNA vaccines, the RNA vaccines, which I'll talk about later, or vaccines which have used an adenovirus vector, which I'll also talk about, have really been based and focused on this spike protein. So why was the spike protein originally chosen as the basis for most of these vaccines? So the receptor binding domain of the spike protein, so this is the part that actually sticks to the ACE2 receptor, it's thought that if you have an immune response that targets that binding, that will prevent entry of virus into host cells. And if you can prevent virus from even entering into host cells, particularly the cells in the throat and in the nose, then you have the potential to prevent any sort of infection, including asymptomatic infection. And if you prevent infection, you interrupt transmission and get this herd immunity that we're also keen to try and achieve. Other spike protein epitopes and other parts of the spike protein may also be important though, because they may contribute to stopping the virus attaching to the host cells, may contribute to neutralization of the virus through antibody, and may also prevent fusion of cells once the virus has attached. The benefit of a vaccine targeting multiple parts of the spike protein means that mutations are less likely to lead to evasion of immune responses. So if you have a vaccine just targeting one part of the protein, it's relatively easy for the virus to mutate and avoid vaccine-induced immunity. Whereas if you target multiple protein epitopes, that could be an advantage. And most of the vaccines we have right now that are being used target multiple spike protein epitopes. So just thinking a little bit about what we're trying to achieve and sort of a bigger picture, for the most part, vaccines are trying to reproduce immunity you get after natural infection without the infection. And so this just shows what, at a high level, the sort of immune responses that can occur at the extremes after infection. So I'm going to start on the left-hand side here, as what has been referred to in this picture as a dysfunctional immune response. So these are cells in the lungs. You have infiltration of T cells to kill the viral infected cells, macrophages, which are also cells in the immune system that kill the virally infected cells. You have a lot of cytokines being produced in the cytokine storm, which has got a lot of attention. You get edema, so swelling in the lungs. You can worsen pneumonia, and essentially a lot of inflammation and damage to organs. And this is inflammation and damage being caused as a result of the immune response, not necessarily as a result of the virus. So this is clearly not the sort of immune response we're trying to achieve through vaccination. What we'd like to see is here what's referred to as a healthy immune response. So you have virus here being neutralized by antibodies. Any virus that's got into the cell is being prevented from being released. You have T cells here that are responding in a very nice way to eliminate infected cells without causing excessive inflammation, virus being neutralized by antibodies, very little lung damage. And so ideally this on the right-hand side is the type of immune response we're trying to achieve, and you can see here that antibodies are thought to be critical to this immune response. So thinking a little bit more about how antibodies work. So here a little cartoon again of the virus with the spike protein on the surface. And so if you have antibodies which prevent binding of the virus to the ACE2 receptor, the virus doesn't even get into cells. You can also have antibodies here which prevent fusion once it's within the cell, and release of the virus genetic material. So even if the virus is in the cell, it prevents further propagation of the virus. So again, this is what we're hoping and expecting antibodies to do, and these antibodies hopefully being induced by vaccination. And the evidence that antibodies, and particularly antibodies that are able to neutralize the virus, has really come from a study of non-human primates. And this is a study that was published fairly early on, but is still very highly cited. And essentially this shows, using a couple of different methods, that the more neutralizing antibody is produced in non-human primates, the less virus they have, whether you look in the bronchoalveolar lavage, so in the lungs, or in the nose. The more neutralizing antibody they have, the less virus they have. And on the right-hand side, shown in a slightly different way, that rhesus macaques here, those with complete protection had very high levels of neutralizing antibody, and those with partial protection had slightly lower levels of neutralizing antibody. So this really led a lot of extra weight to the theory that high levels of neutralizing antibody elicited by vaccination would protect against COVID-19. But antibody has lots of other functions, and some of that is summarized here on the slide. So we have the neutralization at the top. They also can opsonize viruses and other microbes. So they essentially stick to the surface of the virus, and instead of neutralizing it, they then connect it to other cells of the immune system, like neutrophils, which are able to digest the viruses and kill the viruses that way. So even non-neutralizing antibodies may have this effect. As well as these phagocytes, which can take up and kill the virus, you also have other mechanisms through which antibody can lead to killing of the virus through natural killer cells here. And there's also other mechanisms through things like the complement system, where complement binding to the surface of a virus can also lead to killing and elimination of the virus. As well, at the bottom, you can also get inflammation as a result of the immune response, and this is what we're trying to stay away from. So while it was thought that neutralizing antibodies are important, there's, at least we know from other immunology that other antibody effector functions may also be important. And remember, what we're trying to avoid is potential harm from any immune responses. So again, there's been this theory based on animal models, mainly from SARS-CoV-1, that any antibody that binds to the virus that does not neutralize it may allow it to enter cells that it may not otherwise enter, and essentially cause worsening of infection. So while we haven't seen this yet, this remains a sort of low-level concern. So what happens to immune responses to COVID over time? So first of all, look on the left-hand side here. It's a study published recently looking at total antibody responses on the upper left panel and neutralizing antibody responses on the lower left panel over a period of 240 days after infection. And you can see that antibody levels, whether you look at total antibody or neutralizing antibody, are maintained fairly well over this period of time, only a slight drop-off, which bodes well for any vaccines which are able to produce antibody if antibody is indeed protective. I haven't talked very much about T cells, so other parts of the immune system which are important for protection against viral infections, and we touched on that at the beginning when I mentioned that they in part kill virally infected cells. And you can see here that over a similar period of time, looking at different types of T cells, so CD8 T cells and CD4 T cells, there is again a drop-off, which is slightly more pronounced than you see in antibodies. And in this study, the half-life of the T cell response was estimated to be around 3 to 5 months. And so you do get some drop-off. This is important because depending on whether vaccines are protecting through antibodies or T cells, we may expect the duration of that immune protection to be more or less. So I'm going to just move on slightly to look at what we know about some of the vaccines we currently have available. So some of you may have seen some of these data already. So this is from the New England Journal paper of the phase 3 trial of the Pfizer mRNA vaccine. And over sort of 2 to 3 months after vaccination, you can see here an accumulation of COVID cases in the placebo group in the blue line. And once you hit around 14 days after the first dose of vaccination, almost complete flatline and very few cases in the vaccine group. And that's blown up in this inset here, where you can really see protection starting very early, or within around 14 days or so after the first dose. And here the second dose was given at 21 days, so protection occurring even before the second dose is given. Overall, the vaccine efficacy was 95%, and that's the headline. And if you look in a little bit more detail after this first dose, so if you go after dose 1 to dose 2 before dose 2, overall efficacy was 52%. But that includes this period in the first 14 days when there's no efficacy, and you wouldn't expect any because it's too early for any adaptive immune response. And so all of that 50% comes from this difference occurring in that 14 to 21 day period. And actually, if you look at the data more closely, at this sort of point, you're already getting up to around 90% vaccine efficacy from 14 days after the first dose, even without the second dose, at least for this short period of time. For the Moderna mRNA vaccine, again, you have vaccine efficacy of around 93% after 2 doses, and you can see very similar trends here. The placebo group with accumulation of cases over time, and in the vaccine group, almost complete flatline once you get beyond that first couple of weeks after the first dose. In this study, the second dose was given at 28 days. In this particular study, they did do a bit more detailed analysis of the efficacy after 1 dose, and essentially overall it was 80% after 1 dose, and if you just look at least 14 days after that first dose, it goes up to 92%. So similar data for both of these 2 mRNA vaccines of efficacy of 94–95% after 2 doses, but getting up to 90% even after just a single dose, at least short term. Just to look at a slightly different platform, so this is the Chadox Oxford AstraZeneca viral vector vaccine. So this is a chimpanzee adenovirus vector, which expresses the spike protein, and again, very similar curve. So here they didn't have a true placebo. The meningococcal quadrivalent vaccine was used as a placebo group, and you can see here in the meningococcal vaccine group, accumulation of cases over time and much less accumulation in the COVID vaccine group. And in this particular trial, overall, the vaccine efficacy was around 70%, so slightly lower than the 2 mRNA vaccines. But how does that relate to immune responses? And sort of having taken a bit of time to look at some of the immunologic data from the early trials of these vaccines, this is my sort of very oversimplified summary. So first of all, for the 2 mRNA vaccines, the data are very similar. But in essence, after the first dose, you have some non-neutralizing antibody here in blue, being driven by clearly there's the B-cell response driving these antibodies, and a small amount of a T-cell response as well. Once you get the second dose, you have a little bit more of the non-neutralizing antibodies, a lot of neutralizing antibodies, and expansion of that T-cell response. And I think the key thing to think about here is that if you remember, after dose 1, we're still getting, at least in the short term, around 90% efficacy. After dose 2, it's going up to 95%. So going back to earlier on, the concept that neutralizing antibody is the only protective mechanism clearly can't be true unless you need very, very small amounts of neutralizing antibody that were not really detected in these vaccine trials. So that's a possibility. Other possibilities are that these antibodies that are not neutralizing are protecting through other mechanisms. And I mentioned some of those earlier, or there's a contribution of T-cell immunity as well. For the adenovirus vector vaccine, it's a little bit different. And here, after the first dose, you do get both binding and non-neutralizing and neutralizing antibodies, and probably a slightly better T-cell response than the mRNA vaccines. And again, this expands after the second dose, particularly in terms of the antibody response, where the T-cell response does not seem to change that much after the second dose for this particular vaccine. So slightly different kinetics, lots more neutralizing or some more neutralizing antibody with this vaccine after 1 dose compared with the mRNA vaccines, and slightly more of a T-cell response. And then at the end, you're getting moderate levels of neutralizing and non-neutralizing antibody, not much further expansion of the T-cell response, and ultimately a slightly lower vaccine efficacy, at least in the trials that were done, although we have to bear in mind that these vaccines were not compared head-to-head in the efficacy trials, so there may be other reasons for the differences in the vaccine efficacy. But it leads us to think about what in this immune milieu is actually protective. And is it neutralizing antibody? Are there other factors at play here? Is it different for this vaccine compared with the mRNA vaccines? And I just want to leave with the conclusion that essentially there is currently no defined correlate of protection against COVID-19 disease or infection. As such, the immunological thresholds required for vaccine efficacy have not yet been defined. So it's really important that the data and the samples we have from these trials are looked at further to try and identify what it is that is protective. And the reason why this is really important is, apart from helping us to understand a bit more about protection against COVID-19, it may help us to understand and predict how long protection may last. But also, if you think about, there's still a lot of vaccines coming down the pipeline, and there's a limit to how many phase 3 trials can be done involving 30, 40,000 people, which is the current, where we are at with the current trials. And ideally, what we want to get to is something in the immune system that we can measure, that we can say, if you have this level of antibody or this level of T-cell immunity, then we think that indicates protection, to enable us to do trials for future COVID vaccines not involving tens of thousands of people. And I think it's really important that we have a clear pathway to licensure for other vaccines coming down the pipeline. With this current uncertainty, though, I think we have to be open to consideration that these correlates of protection and the mechanisms of protection may be different for different vaccines. The mechanisms may be different after one dose compared with after 2 doses for these vaccines. I think it's highlighted by these mRNA vaccines, where we see excellent protection even after 1 dose in the almost complete absence of neutralizing antibody, which everyone thought was going to be the key mechanism of protection. And it's also important, and I haven't really talked about this very much, but as we hear and as we see about different virus variants popping up in various parts of the world, and as the virus continues to mutate, understanding how the immune response is protecting against infection will be important also to try and think about how the variations and the mutations in the virus over time may impact on vaccine-induced immunity and protection. So I'd like to thank you very much for your time, and happy to take any questions. Thank you.
Hi, just double checking that we're live.
Yes.
Okay, can't see anything on the screen, so I'm just going to go ahead and just say thank you to all 3 of our presenters. Amazing opportunity to hear some wonderful content, and I know we've got some questions. And it's also my privilege to add to our discussion this afternoon, Dr. Rob McMaster, who is going to join me in my moderating duties. So welcome everybody back to the screen. And I think what we'll do is just start. There's a few questions in the chat box that I'll maybe read off. And Rob, I don't know if I read them off and you want to direct them to whom you think might be the best person to respond. Does that work for you? Rob, can you hear me?
Yeah, that will work fine.
Okay, good. Sorry. Thanks. Thanks very much. So I'm just going to read this first one off out of the chat. How do vaccine developers make sure that preclinical evaluation does not eliminate vaccine candidates that could have been efficacious in humans or effective in humans? We often see the opposite of this, in that the vaccine candidate appears effective in animal model, but not effective in humans. This is a long question, just about done. Therefore, there are probably candidate vaccines that appear ineffective in the animal models, but effective in humans. Who do you think, Rob, would
Oh, I
take
think
that
Alison,
question?
that's a good one for Alison.
Perfect.
Yes, hi there. That is a great question. Typically, we want to see effectiveness in 2 different animal species before considering moving the vaccine to human clinical trials. I think with COVID-19 especially, many of the major vaccine candidates were evaluated in non-human primates. So they skipped even typically we would do 2 small animal models. They went right to the non-human primates to make sure that they were more comparable to humans. And I think that gave them an advantage. Of course, there was a slight, because everything was more, was done in parallel for COVID-19 vaccine development. Several of these preclinical studies were done at the same time, and I think Health Canada didn't require a 2-animal model before reviewing applications. So for COVID-19, slightly different.
Excellent, wonderful. Thanks for that response. I'm assuming there's no other additions from anybody else. I'm going to take that as a no. And the questions are piling in. So I'm going to do this next one. I suspect, Dr. Calvin, this one's probably for you again, but we'll let Rob direct. Are there preclinical data that show in the DNA-based vaccine, the DNA will not be randomly incorporated into the mammalian chromosomes? DNA plasmids are known to sometimes randomly incorporate themselves into mammalian chromosomes and cell lines in vitro.
I don't remember the papers that Manesh was referencing, and that was probably a good place to look if there was any investigation into that. But I don't think that that would be a big concern, but maybe he would have more insight since he referenced that paper in his talk.
Excellent.
Yeah, thanks, Alison. So I don't recall specific analysis around that. There may have been some data even earlier than these specific vaccines, to be honest, looking at some of those issues. But I think, as Alison said, the nature of most of these DNA vaccine platforms are that they don't incorporate into host mammalian DNA.
All right, excellent. Here's another one. What is the reason not to use synthesized ACE2 protein motifs to competitively inhibit viral binding to ACE2 on host cells? Is it due to the side effects of using ACE2 motifs? Who would like to take that one?
Well, I could take it, actually. If you look at Josef Penninger, has a study using ACE protein, the whole protein, as a block for virus. So it's actually using that to, I guess, saturate the virus in ACE receptors so it can't bind. It's a soluble receptor, can't bind to the cell. And I believe it's in phase 3 studies at the moment, mainly in Europe. I don't think in Canada at this point. I haven't seen, he had a case report on the last meeting we were at. So it is in trials, and it's proceeded to phase 2. There's phase 3, it must be showing some effect. There was concern that by having it's an active protease, it has a whole effect on blood pressure, is that by introducing an active molecule that you actually disturb the blood pressure across the body system. And it showed no evidence for that. So it was reassuring that it's acting as a decoy for the virus. And I think we look forward to those results coming out. If you've heard him talk, that was from SARS-1 that we brought up for the current COVID. And it could be an alternative treatment. I think the other advantages of it is it can also hopefully decrease the effect on other organs. We know that serious disease is a multi-organ disease, that it could stop the virus from affecting kidneys, GI tract, etc. So there are studies with that already in progress.
Wonderful. Thanks, Rob. So we have another question here, and maybe Rob, again, I'll ask you who you think can best respond to this one. How can subpopulations make decisions on getting a vaccine if we don't provide data? Are there phase 4 trials working on this? If not, how are pregnant and breastfeeding women, immune compromised patients, et cetera, supposed to make decisions? That's a great question. Who wants to take that one?
I think that's Darby's question.
Okay.
Because that's come up a lot, hasn't it?
Yeah,
Yeah.
I can give that one a try. Maybe there's some biology going on as well that can be supplemented by our other speakers. But I think is a incredibly important question, and one that a lot of people are nervous about, thankfully, because we have shown such good efficacy in the primary phase 3 trials. So that there is a lot of interest in getting the vaccine. So number one, we're in a really good place to be asking these questions. So they're not phase 4 trials ongoing. I'll say right off the bat that I think both Pfizer and Moderna are undertaking de novo trials, or small phase 2, if you will, trials in pregnant women to understand the immunology and the potential protective efficacy in women, in pregnant women. So I do expect there to be some results in the near future regarding those. If any of the other speakers have any other information about that, that would be fantastic. But in general, I mean, the question is broader than that. It's about all the subgroups of people, of vulnerable people who are at risk for more severe disease, immune compromised, or with other chronic conditions. And those will be evaluated as we go. The number one priority was to see if we had something that worked. And there's going to be a lot of drilling down into the data. They enrolled 40,000 people into just the Pfizer trial. They will be digging into that and looking to find as many subgroups as they can identify to determine what extent they need further study and to what extent there's a possibility of rolling out treatment there. But I have to suggest there has to be some level of patience there. Analyzing this data is incredibly challenging. It's a huge amount of information. They're still finalizing the full safety database on the 40,000 people. That's going to take a little bit of time before they're able to start teasing out potential effects and safety signals within subgroups. But I guarantee you the extent to which they can, they will. And the extent to which they can't, I expect that there will be further either industry or government-sponsored research into important subgroups with people with chronic conditions. So I don't have a good answer,
Yeah,
but,
really
they're going to try and
question.
seek it out.
Thank you,
Yeah.
Yeah, I can add to that a little bit if you want. So I think that, again, I think these are really important questions. I think, the decision around which populations to include or exclude in the initial trials is a difficult one, especially when we're in a pandemic and we're trying to be as inclusive but as safe as possible. And we could probably argue all afternoon whether the lines were drawn in the right place, but they were drawn where they were drawn. So most immunocompromised people were not included and pregnant people were not included. I think from the phase 4, while we know that there are people from these groups that are being vaccinated and have been vaccinated because they're being called because they're in one of the other risk groups, whether they're healthcare workers or dependent on age or whatever. And there are some, studies that are being planned to evaluate safety and effectiveness in those populations. And I have no idea right now what the size of those trials will be, but there are those studies planned. And as Darby mentioned, there are also sort of de novo phase 2, phase 3 trials that are being planned in those populations as well. So I think over the coming months, we'll have a combination of some phase 4 data supplemented with the phase 2 and 3 data as well.
Wonderful. Thanks very much. Any other comments?
Yeah, I could speak a bit to the pregnant women, in that it's typical for pregnant women to be excluded from phase 1 and to phase 3 clinical trials for vaccines. This was done for the influenza vaccine in 2009, and was also done for both of the mRNA-based vaccines. But for both of these examples, to review or to get some preliminary data on pregnant women and their uptake of the vaccines, what has been done now, and which was done for 2009, is that some of the data was gone back and reviewed of women who didn't know they were pregnant when they got the vaccine, and pregnant and women who became pregnant shortly after getting the vaccine. So when mRNA and Pfizer's vaccines were first approved, pregnant women were advised not to get the vaccine. But now that this data has been reviewed and there doesn't seem to be any issues in pregnant women, now it's being advised that they can discuss getting the vaccine with their doctors.
Wonderful. Thank you, all three of you, for answering that. Really appreciate it. So we've got another question. This one's for Dr. Thompson. Are there any statistical considerations for phase 1 clinical trials, specifically regarding participant enrollment?
So regarding design, these are usually designed without strong considerable statistical considerations with respect to powering a study to detect certain endpoints or rule out safety signals or that sort of question. So in general, the level of statistical planning for phase 1 really pertains to ensuring that you're capturing the data necessary to enable phase 2, that you're collecting the relevant data and elements that will allow decision making, and to ensure that you're starting to expose or planning to expose and move towards the target population. As you'll see in some of if you read the paper I described, there's often several phase 1 studies. I mean, we even just talked about some of the subgroups where we might need to restart. We have done healthy adults. We might move to people with an immune condition, or then children with that are immunocompromised or another subset. So really the planning is foresight beyond the phase 1 trial, not necessarily for powering or design of the phase 1 itself.
Excellent, thank you very much. Next question. Similar to the previous question about DNA vaccines, is there any data out there showing evidence of adenovirus vector-based vaccine DNA incorporation into the host's chromosomes? Now, Rob, who do you think is going to need to answer that one?
I mean, Alison was talking about some of the vaccine development. I guess the question is there transfer of DNA from a DNA-based vector?
Right. So using the viral vectored vaccines, typically the ones with the adenovirus-based vaccines that have DNA encoding, say, the spike protein within the adenovirus. As far as I know, there isn't any data out there indicating that there is recombination, especially because this is incorporated within the viral genome and not so much a free piece of nucleic acid. But I can't speak to anything beyond that.
Okay, great. Thank you. Here's another question, and maybe Rob, again, I'll ask you who you think best would want to respond to this. So given that the Oxford vaccine is only 70% effective, do you see it getting approval by Health Canada? And if so, when? Also, can you comment on the second dose being a half dose?
I think Darby actually had some data on percent.
Yeah, I mean, you were asking who wanted to answer it. I don't want to answer it, but I suppose I can.
Well, you're not Health Canada, so you don't have to.
Yeah, exactly. I do not speak on behalf of Health Canada. So
that's tomorrow morning. There's Health Canada on.
Yeah, exactly. I mean, we can't speak to what's going to happen in the future because as we already know, there's several subtypes and variants, and we don't know the extent to which all the vaccines are going to be effective against all the subtypes and strains. And it's very possible that there's a scenario where we might be requiring several vaccines for long-term, for conveying immunity well into the future years that come. With the COVID season coming every year, we might need several different approaches because, like I said, the context is variable. Today, it does not seem like there's a lot of evidence to approve it. I don't know what tomorrow looks like because, ask me what January 28th would have looked like in 2020. I wouldn't have been right. So I can't speculate to what might happen, but it's possible. I think Manesh or Alison probably have some other insights there.
Yeah, I could just make a brief comment. I mean, I think that just taking a step back and looking at the bigger picture, I think 6 months ago, if you'd offered people a vaccine that was 70% effective, we would have jumped at it, right?
Absolutely.
So, you know, the 90% we've seen with the mRNA vaccines was, I think, at least to me, a little bit of a welcome surprise. And, you know, I think Alison highlighted really well that if we're going to vaccinate the planet, we need multiple vaccines. We're not going to do it with these just mRNA vaccines. And we don't know how things are going to play out. We don't know anything about long-term duration of immune protection. And while you might speculate that it would be better long-term if the initial protection is higher, I don't think you can say that for sure. So I think, you know, I also don't know what Health Canada is going to do, but, you know, at the end of the day, that Oxford AstraZeneca vaccine did sort of meet the minimum requirements that had been set out by regulators at the start in terms of the minimum efficacy that was required, and they've done that. So, you know, and there are other factors to be considered, such as the, you know, storage, transport requirements and cost and those sorts of things. And clearly the cost isn't something for the regulators, but I think for the rest, I think it's their job to decide whether the vaccine is safe and effective and sort of does what it's supposed to do. And then how it's deployed is really goes onto the national immunization technical advisory groups and other policymakers. I don't think that's necessarily just a question for the regulators.
Wonderful answers.
I actually had a related question is, you know, on really clinical trial design, is that when we have this mass immunization, say in Canada, everyone should be immunized by September, how are we going to conduct trials on new vaccines? Say if you want a Canadian vaccine, you know, there's one announced yesterday that's coming through. Alison talked about several that are in development. What kind of are we going to think about for clinical trial design to actually assess, particularly say phase 3?
Yeah, so yeah, all really great comments, and I don't know, so I do have a question actually in a bit of a different vein for all four of you. Robin, I'm not sure if you wouldn't mind answering this too, but in light of the conversation this morning around learning health systems, and as well, we're going to carry on some of that discussion in a different session this afternoon, I'm wondering if each of you wouldn't mind thinking about what you would think is important for health systems leaders to understand about vaccine research. So that sort of that integration of knowledge into practice type of idea. So don't know who wants to tackle that first, but is there a key message from a decision maker point of view that you would elaborate on about vaccine research, either from a COVID perspective or just broadly speaking from a health systems perspective?
Well, I think one aspect is, you know, supportive research in general. If you look at the lipid-based vaccines, the Moderna and the Pfizer, you know, a lot of that technology and encapsulation was developed at UBC with Peter Collis's group 20 years ago. It was never, even though it wasn't contemplated really for a vaccine, it was more in gene therapy. And it was really, we call it, you know, fundamental or discovery research that, you know, the companies were formed out of it, and all of a sudden it's become such a powerful vaccine. So I think it's not just focusing on applied research of vaccine, it's also supporting different aspects. If you look at Manish's work on the immunity, you know, if we didn't understand the immunity, we wouldn't know what we were looking at. And a lot of that is really fundamental studies on both human and experimental animals in understanding the immune system. There's lots of questions on second, you know, prime boost, et cetera. Well, we know a lot about the immunology, you know, not necessarily from a vaccine development stage, but it's all important to you need to bring all that information together to understand a vaccine. So I think the message is we need to support research across the board.
I'd like to pick up on that really quickly, because that's essentially the way I was going to answer your question, Rob, about how would you be able to test things in the future. Well, it's about integrating research into delivery of care. It's about understanding that, you know, we know a lot, but it's all evidence. And we have to continue to accumulate evidence to answer new questions, subgroups, pregnant women, boosting, half doses, storage, stability, continually answering these questions. And particularly in Canada, one of the best ways to do that is to integrate research into how we're providing therapies, vaccines, and care to patients.
Yeah, I would agree. And I would sort of add to that. I would say, I think historically this sort of boundary between clinical care, either at an individual level or a population level, and research is an artificial one. I think, you know, we're only going to have high quality healthcare if we have research as part of that. I think what there does need to be, though, is an ongoing bi-directional conversation, collaboration between the people who are involved at the front end, whether that's policymakers or clinicians, and the people who are doing the research, to make sure that the researchers know what are the main questions. I don't think any of us, you know, any people involved in vaccine development want to spend weeks, months playing with vaccines in the lab that no one's going to use. But at the same time, once they have stuff that is ready to go, I think the policymakers need to know that, you know, to get it into policy as soon as possible. And I think actually the last year and what's happened during the pandemic, things have improved, and I would hope that in the future we can build on some of those collaborations that have been made.
The other thing to add there is to include patients, patient advocates, and specific advocacy groups in the research process, because even if the scientists, the basic research and the clinical trials research all point to certain answers, the willingness to uptake some of that information at the patient level, their comfort with risk, their comfort with the knowledge of their diagnosis or their treatment regimen is important, and helping them understand that you know what, your doctor, the trials, we actually don't know everything. We have really good evidence to support things, and if you're uncomfortable with this, and we determine that the huge segments of the population are uncomfortable with things, that's a direction for research to further understand and get people confidence in how they're being cared for and their active role in it.
Such great points. Dr. Calvin, do you have anything you want to
so
add?
I really want to stress, especially in our current situation, both in the pandemic and where we are with our vaccine procurement and deployment, is that we really need to strengthen what we can do in Canada. I think the pandemic highlighted, you know, where our deficiencies were, to both our ability to do high containment, the few researchers and research facilities that were able to do that, to who is actually able to develop a vaccine platform, to who can scale it up, and do we have that capacity to do it? You know, I think we do a great job with human clinical trials and clinical evaluation, but we don't have that groundwork to really base our own vaccine on. And without that, then we're dependent on the rest of the world, which has put us in this vulnerable position. And relatedly, I think it's also really important, just as the other panelists mentioned, to have good communication, not just to the government stakeholders and policymakers, but also to the general public and our communities, and not just, you know, the everyday large city communities, but right down to our uniquely Canadian communities that are remote, and make sure we're getting that information out there so that we are protecting the people that we're working so hard to develop vaccines for. So those would be my big messages.
Well, that's a wonderful way to end this session. We've only got about a minute left. There were a couple of extra questions that we'll make sure get out to you that you can respond to in writing. Just want to take this opportunity to say thank you very much to all the panelists for the presentation. Thank you to the audience for listening and for the amazing questions that we saw. Really appreciate all of your time, and wish you all the best. Please stay safe. Thank you.
I think it's been a terrific session. I've learned a lot in the last hour, and really look forward to continuing our dialogue. And thank you, everybody. Thanks to hear the speakers. Excellent talks.
Yeah.
Thank you all.
All right.
Thanks.
Go let your dog.
She's been uncharacteristically silent, although she did walk by and look at me grumpily a few moments ago because I think he was making too much noise.
Good.
Wonderful.
Thanks, everybody.
Thank you.